community to welcome you at this webinar and make maybe a very short introduction to what VAC4EU is, for those who don't really know yet what VAC4EU is. It is Belgium, well, it's an international non-for-profit organization that has been created under the Belgian law. We are three founders, the Professor Sturkenboom and Vincent Beauchot and myself. My name is, as Miriam just introduced me, Patrick Meilly. And as a Secretary General, I'm very pleased to introduce you to VAC4EU, which is a partner in the Access project, so providing governance network of site and infrastructure for design and conduct of post-licensure vaccine study, which is exactly one of the main goal of VAC4EU. We have a lot of members that are essentially public health organizations in Europe and universities. So that is the way where members conduct any of the proposed studies for different study requesters according to VAC4EU governance model. So for those who are really interested to work with VAC4EU in the context of studies, I'm inviting everyone to contact Secretariat at vac4eu.org, and then we'll proceed with the development of possible collaboration and studies all together. The next slide will be concerning the background and setting of the Access project, and Miriam will take it over and explain you what is exactly the role of VAC4EU in this Access project. Miriam?
Thank you very much, Patrick. So my name is Miriam Sturkenboom. I'm one of the founders of VAC4EU, but also coordinating the Access project. So together, and it's co-led with University Utrecht with Olaf Clement. So this project is a project that was tendered by the European Medicines Agency, and the Access project partners, which are 22 in total, are developing various template protocols to prepare Europe to monitor COVID-19 vaccines. They are also assessing capacity throughout Europe, going very broad to all member states to see whether anybody would be interested and is capable to participate in the any of the proposed studies. So these activities are funded by EMA until December, and by that time we will have protocols written for background rates. This was already delivered and is available on the UPAS register publicly. We will also write protocols for effectiveness, coverage, and safety. Today, the focus will be on the methods of the safety studies that we are proposing. So it will not be the entire package of what Access is doing, but just focusing on the safety. Next slide, please. So what we will discuss today is safety monitoring study methods with different types of data that are proposed and that will be delivered to EMA this month. So the purpose of today is that we, as VAC4EU, really would like to open the methods discussion to the audience, so to have an exchange, a scientific exchange. This is in a webinar mode, so if you have questions, please use the questions box to put your questions. After each of the proposed methods, we have a 5-minute discussion. We have in total 3 presentations, so a 10-minute presentation and 5-minute discussion, and the time remaining at the end we will also use for a general discussion. This webinar will be recorded and shared publicly on the VAC4EU website as the previous webinars are. Next slide, please. So just a bit of an overview, because we are going to dive very quickly, deeply in methods. So if to design vaccine safety monitoring and to have Europe prepared for the monitoring of the COVID-19 vaccines, there is a variety of methods, of course, that can be utilized. And some of some of the activities can actually be done already now before we have the vaccines actually being rolled out. One of the, and that is the background rate. So calculation of background rates of adverse events that we may think may, if they would arise, would be causing a problem. So we want to be prepared in knowing how often they occur without the vaccine, so they can be used to interpret signals. The Access project wrote the protocol to do this and is currently calculating background rates on 38 events in 7 countries. And this protocol is publicly available. So this is already ongoing. Then there is safety monitoring that can only be done after the vaccine has been rolled out. So we cannot yet do it, but we can be prepared to do it. So there is, I think, 3 key aspects: signal detection, rapid assessment of signals, and evaluation of safety concerns. So as a routine pharmacovigilance measure in the RMPs, there's of course always the spontaneous reporting, which is already existing and maybe enhanced or speedier, but that is an existing tool. So we are not working on that. What Access has been working on is 5 different pieces. It's in the signal detection cohort intensive monitoring with patient reports, and this will be presented by LAREP. We also have some methods that are using EHR data, and that is additional signal detection on EHR, and this will be presented by Caitlin and Corinne. Also, in a rapid assessment of signals, you would want to use EHR data to quantify impact. That's in the same protocol from UMCU. And then there are evaluation methods of safety concerns. So there we talk about really hypothesis testing and quantification studies. And this protocol, there is a protocol that can be done in Sentinel hospitals, and this is being developed by RTI. And then there could be options to do studies, cohort and case-based designs in EHR data. So this is the menu of what you will be looking at, the last 5 items actually. This is what will be presented. So maybe next slide, I would like to give the word to Lynn to present the intensive monitoring of COVID-19, which is really the, focusing on signal detection. Lynn Nurse is working at the Netherlands Pharmacovigilance Center, LAREP, together with Agnes Kant, and they have been developing these methods and the protocol for it.
Yes, thank you very much for introducing us, Miriam. Yes, so next slide, please. So within our task, we have 2 deliverables. One is a protocol in which we design a European intensive monitoring system to generate knowledge on adverse events following immunization, AV, of COVID vaccination. The other deliverable will be a report to assess the feasibility of the proposed intensive monitoring system in Europe. Next slide, please. So the aims of this intensive monitoring will be to describe the pattern of patient-reported AFIs, so the frequencies and the course of disease. And we'll do this both on the national level and the European level in near real time. And the AFIs will include expected as well as unexpected AFIs. This is the primary aim, and the secondary aims will be to describe differences in patterns of AFIs between the various vaccines used and between the different countries. And furthermore, we aim to identify possible risk factors for adverse events following immunization. Next slide, please. So more specifically, the research questions will be: what are the reported AFIs for each vaccine and in each country? What are the frequencies of these reported AFIs for each vaccine and in each country? What is the course of reported AFIs? So then we mean the latency time, the outcome of the adverse event, and if the patient recovered, also recovery time. What is the impact of the reported AFIs, and are there any possible risk factors for AFIs, and which are these risk factors then? Next slide, please. So the study design is a prospective cohort study, which will be single-armed. So the observed AV incidences in our intensive monitoring, they can be compared to the background incidences that are currently being calculated within the other access deliverable Miriam just explained. So in the different countries on a national level, the data will be prospectively collected in near real time directly from a cohort of vaccine recipients. And then furthermore, the common core data from the different countries will be pooled and analyzed at the European level. Next slide, please. So this, so in order to get homogeneous data and to be able to pool all the data from the different countries together, we'll do our best to really harmonize the data collection between countries. So therefore, we have a list of core questions, a minimal set of core questions that we want to ask in each country so that we collect exactly the same information. We also will also set a minimum set of follow-up periods upon each vaccine dose. So we'll collect data at day 0, 7 days after vaccination, and then 30 days after vaccination. And then later on, we'll probably also have follow-up periods, and we'll then aim to have the same follow-up periods for as many countries as possible. To have as little variation within that, these follow-up periods. Then about the data collection tool. So at LARAP, we are working on a so-called LARAP Intensive Monitoring LIM web-based app, the LIM web app. So we will make this available to other countries, but countries can also use their own tool if they already have this available in their own country. And they can still participate in the joint intensive monitoring. So if the country uses the LIM app, the data will be automatically stored centrally. And each country can access the data originating from their country, so the full data. But at LARAP will pseudonymize this data so that it can also be pooled together and analyzed at the European level. And then countries that use their own app, they will store their data locally, of course, but then we'll try to harmonize the aggregation of their data. For example, on the AV frequencies. So last of all, we have the joint statistical analysis plan, which will include standardized queries to, for example, calculate these AV incidences on a national level. And then we can pool the aggregated data together on the European level and produce 2 weekly reports on these frequencies of AVs, for example, and to visualize this data. Next slide, please. Yes, so these about the sample size. This is a very rough estimation or order of magnitude we are now thinking about, but of course it also depends on the size of the countries that will participate. But assuming that we would have 6,000 participants with complete data in one country, this would allow us to detect the AV with a frequency of 1 in 2,000. And if you would then have like 5 countries like this, on the European level, we would have 30,000 participants, and we could detect AV with a frequency of 1 in 10,000, just to have an idea. Next slide, please. And then this slide summarizes the basic core data that we're aiming to collect. So this list is still a bit in progress. But anyway, we'll collect background information on the participant, like age and gender, height and weight, information on pregnancy, if they're also pregnant women being vaccinated. We'll collect information on the medical histories, for example, whether people were already infected with SARS-CoV-2 or whether they had the COVID disease. We'll ask about allergies and chronic disease, as well as co-medication. Then we'll collect information on the vaccination date and on the dose of the vaccine, if we would have a vaccine with multiple doses. We'll collect information on the vaccine brand and batch number as well. So this can be obtained either via the vaccine recipient and or via a linkage with the vaccination register. So this will depend on the situation in a different country or in the different regions of the country. And then most importantly, we'll collect information on the adverse events that are attributed to the vaccination. So these will include closed questions on, for example, injection site reaction, and then with different sub-questions, for example, warmth or redness of the injection site. Another closed question on fever, with a sub-question on temperature. And then we have additional closed questions on other very common APHIS, which we are not officially, which we don't know yet. We'll can base this on the SMPC, for example, of the vaccine. And then in addition to this list of closed questions, we also have an open question on other events that are attributed to the vaccination. And then for each of these reported APHIs, we'll ask about the course of reported APHI, as well as the impact. So with impact, we mean whether the participant sought medical assistance or whether he or she was hospitalized, and also what the impact was in general. Next slide, please. Oh, yeah, so thank you very much for your attention. Please let me know if you have any questions or remarks.
Thank you very much, Lynn. Are there any questions? We have some time for clarifications and questions on this. And is there anybody who would like to ask a question? I see from you said that Florence Costa had a question about ethics approval in each country. Can you comment on that, Lynn?
Yes, so each country will be responsible for for that. Yes, yeah.
Okay, is there any other question that people have? You can raise your hand or put it in the questions.
Yeah, I see multiple hands raised, but let me check if I can unmute. Yeah, I see a hand raised by Elena Kromava. I unmuted you now, so you can ask your question.
Thank you for this presentation. My question is about a link between the patient-reported data and the medical confirmation of the diagnosis. It may be possible, maybe in a single country, but maybe not in all. How do you anticipate to link, especially for, let's say, for hospitalizations and for serious adverse events, you know, what patient reports with, I would say, the confirmation of the diagnosis at the hospital or GP level? Thank you.
Yes, thank you very much, Elena, for your question. So in general, we do not anticipate that there will be many serious adverse events reported within the intensive monitoring, but they would rather come in via the spontaneous reporting system. But of course, if there would be more serious adverse events, then we'll do a follow-up. Yes, there will be assessors that will contact the participant if they provided, how do you say it? If in the informed consent they check the box that they could still be contacted. So and then we could do this via the patient. Does that answer your question?
Yes, thank you.
I think we have time now for one more question, and then we have the two other presentations. But please, for people, if you have a question, also write them in the questions. They will be recorded, and if we do not have the time to address all of them, we can come back to you afterwards.
Yeah, I see a hand raised by Marie-Helene Gullet. I unmute you now, so you can ask. We do not hear you yet, so you have to unmute yourself as well. Yeah.
Okay, I think it's good now. Thank you very much. My question was regarding the method you intend to use to collect data about vaccinees, whether they have a IFI or not. Is it going to be a vaccinee registry in Europe or per country? Or is there any plan to get this information?
Yes, that's a very good question. Thank you very much. So yeah, this is now depends on the country or even the region within the country. So I know, for example, here in the Netherlands, yeah, it's still a work in progress, the vaccination register. But in other countries, it might be already available. This I'm not sure. So this is one way to collect this information, of course. If the patient, him or herself, get the information on which vaccine they got and about the batch number, this is also an alternative route, or maybe we should try to have both routes at the same time, so that we can both link the patient-reported data to the vaccination registry and as well as collecting information from the patient, him or herself.
And the idea is also to have a denominator to calculate the frequency and compare with the background incidence. We will need to know how many patients receive this vaccine.
Yes, if I may, yes. Lynn. Yeah, so there is another protocol within the Access project that will be focusing on calculation of exposures and coverage. And it is awaiting a bit to get more information on how the vaccines will be rolled out, which is currently in many countries still in development. So SAGE has recommendations, and now there will be country-specific implementations. So we are aware, and that's important, and it's work in progress. So I would, in view of the time, I would like to maybe close this part now and continue with the next presentation. So this was a protocol really on the ability to detect signals. Now we're going to look at a protocol that will be more able to assess safety issues. And this has been developed by RTI Health Solutions, and Alison Kawai and Alejandro Arana will be presenting.
Yes, hi. Thank you, Miriam, for the introduction. If you turn to the next slide, please. As Miriam mentioned, the task at hand that RTI is leading is to develop a template to provide guidance to study the safety of COVID-19 vaccines using a retrospective hospital-based self-control design, and this work is currently underway. And the template is intended to serve as a starting point for investigators to develop a full study protocol. The template is being designed with flexibility in two ways. It can accommodate many currently unknown aspects of COVID-19 vaccines, including the number of vaccine doses that will be given, the dosing schedule, and the rollout of the vaccine in terms of when and who will receive the vaccine. The template also incorporates flexibility to assess an array of different acute outcomes with short latencies, including both pre-specified outcomes and also safety concerns that may arise during development and after the vaccines become available for use. Today, I will give a brief overview of the objectives, methods, and study design of this approach. Next slide, please. So the primary objective of the protocol is to assess whether there's an increased risk of adverse events following specific COVID-19 vaccine products. And the secondary objectives are to assess these risks by specific subgroups of vaccines defined by a platform or component such as adjuvant, and to also assess the risks by subgroups of patients defined by clinically meaningful factors such as age, sex, and comorbidities. Next slide, please. The study will incorporate data from several hospitals, sentinel hospitals across Europe, and it will use a self-controlled design. And for those who are not familiar with who are not familiar with self-controlled designs, the main strength of these designs is that they implicitly adjust for measured and unmeasured confounders that remain constant over time, such as sex and socioeconomic status. These designs are often used in vaccine safety research to examine acute outcomes with short latencies. And they also, another plus of them is that they typically do not require external controls, although variants of the designs are available to adjust for temporal trends in either exposure or outcome. And 3 types of self-controlled designs are considered in the template, with the preferred approach, the case crossover design and case coverage design, and the less preferred approach is self-controlled risk interval design. And I'll describe these designs in a moment. Next slide, please. The first study design under consideration is a case crossover design. This design is case-based and can be thought of as a self-matched case control study. In this design, cases of an adverse event of interest are first identified. Then one assesses whether the odds of vaccination in an exposure window preceding the adverse event is greater than in a comparison window. In this example, the exposure window is shown here in yellow and the comparison window is shown here in green. The main strength of the design, and this is why we selected as a preferred design, is that no follow-up after events is needed. This is particularly important in our setting of hospital-based studies because we anticipate that it may be difficult to follow up patients after they are discharged from the hospital for the adverse event. 2 additional strengths are that the reverse causation is avoided because exposure windows are before the event. And also, the design does not require external controls. Some key limitations of the case crossover design is that it is susceptible to time variant confounding. However, if data are available, they can be adjusted for. These time variant confounders can be adjusted for regression modeling. Perhaps the most important limitation of the case crossover design, however, is that the design assumes that the propensity for vaccination for a given individual is constant over time. And this is a key consideration. This may not be the case, particularly with COVID-19 vaccines, given that in many settings it is anticipated that vaccines might be first available in high-risk populations and more widely available to other populations later on. Next slide, please. So with this in mind, with this limitation that I just mentioned, the case coverage design was really designed to overcome the key assumption of no temporal trends in exposure for the case crossover design. The case coverage design is a variant of the case crossover design that uses vaccination rates in a reference population to adjust for time trends in exposure. It examines whether each case was more likely to be vaccinated during the exposure window as compared to similar people defined by confounders such as high-risk conditions or age. And like the case crossover design, the case coverage design has the benefit of not requiring follow-up after events, but unlike the case crossover design, is able to adjust for temporal trends in exposure. The main limitation of the design is that data and vaccination rates over time or coverage rates over time are needed in the source population for cases. These data may not be available in all geographic regions. Another limitation is that the temporal trend in vaccination in cases is assumed to be the same as in the reference population. This can be mitigated, however, by matching on confounders. Next slide, please. Because, as I mentioned, coverage rates may not be available in all geographic regions over time, a third approach is necessary. This is a less preferred approach, which is a self-controlled risk interval design. This is a variant of the self-controlled case series. For those of you who know that design, it includes only vaccinated individuals, and it follows a cohort logic. And it compares the risk of the outcome of interest in a pre-specified risk interval to that in a self-matched control interval. In this example, the risk interval is in red and the control interval is in blue. While a control interval could in theory be selected to occur prior to vaccination, we have designed the study to use a post-vaccination control interval. The main strength of the design is that it avoids reverse causation because a post-vaccination control interval is used, and it notably does not require the absence of time trends and exposure, unlike the case crossover design, as I mentioned before. Two key limitations is that it's subject to time-varying confounding. And this is key because, as I mentioned before, we anticipate that covariate information following hospital discharge may not be available to adjust for regression models, which is another reason why this is a less preferred approach. Next slide, please. Now that we described the overall study designs, I'd like to go a little bit into the outcomes and exposure assessment. I won't read it here in full, but here's a listing of the adverse events proposed to be studied with the self-controlled designs. We started with a list developed by the Access Project as a starting point, and we then excluded any events for which the risk interval is not well defined, or that for which there might be a long latency between symptom onset and diagnosis. Next slide, please. In terms of COVID-19 vaccines exposures, as I mentioned before, the primary analysis will be done by specific vaccine product, and therefore it'll be necessary to have information at the product level if more than one vaccine is available in a geographic region. And vaccinations and dates of vaccinations will be obtained from existing documentation, such as patient vaccination cards, medical records, immunization registries, or primary care records. For this study, patient self-report of vaccination will not be acceptable. Next slide, please. An important design consideration that I'd like to mention here is defining the risk and control intervals for the study. And the template provides the guidance for both the risk and control intervals. For the risk intervals, they should be based on biologic plausibility and also data from clinical trials and passive surveillance as available should be used to inform the risk interval definitions. In terms of the control intervals, the key design choices include the length and start. Longer control intervals will lead to greater statistical power, but may introduce more time-varying confounding. As for the start of the control interval, a washout period can be introduced between the risk and control intervals to avoid any carryover effects. And finally, important design decision is that the control interval must not overlap with the risk interval. And this is pretty straightforward for when only one vaccine dose is given. However, as you can imagine, special care must be taken if multiple doses are given. For example, 2 doses approximately 28 days apart. You'd have to think carefully about how to position the control interval if the risk interval is longer, for example, 42 days apart. Next slide, please. So really quickly, just as the conclusion, I wanted to quickly summarize the strengths and limitations of the approach that we're proposing, the hospital-based self-controlled design to assess safety of COVID-19 vaccines. As a strength, the approach avoids confounding by factors that do not vary over time, such as sex and socioeconomic status. It can examine several outcomes with acute onset and short latency, and the approach can be extended to study adverse events with acute onset and short latency. The approach, of course, has limitations in that it cannot be used to examine outcomes with insidious onset or long latency. And finally, the risk intervals must be well specified in order to use the self-controlled design. For both limitations, a more traditional study design might be better suited. And additionally, sensitivity analysis could be conducted varying the risk interval if the risk interval is not well known or established. Next slide, please. And that's the end of my presentation, so I'd be happy to take any questions. And I believe we also have my co-presenter, or my co-author on this protocol, Alejandro, as a panelist. So we're both happy to take questions. Thank you.
Thank you very much, Alison and Alejandro, for preparing this. We would like to focus the questions in for a couple of minutes now on this protocol. So I think Corinne Tamala first. And for all the others that have questions, make sure to write them in the questions box so they get answered even afterwards. Corinne?
I have muted Tamala. Okay, maybe the raised hand wasn't
Okay, so Vincent, do you have a question? Is it...
You're unmuted, Vincent.
Vincent, we cannot hear you.
Okay, then I have a raised hand from Deborah Layton. I've unmuted you. Okay. I don't think we can hear you. I have a raised hand by Wantin Wang. I unmuted you. Could you please try to speak up?
Yes, so my question is, how would you address the potential confounding by COVID-19 and in your design? Thank you.
Yes, thank you for the question, Wanting. So the plan is where data are available on COVID, SARS-CoV-2 is to collect data on it and adjust for it in regression models with time varying as a time varying confounder. And where those, I'll just add, where those data aren't available, the plan is to treat calendar time as a proxy for circulating wild type infection as a time varying confounder, realizing that that approach is less desirable.
Thank you. We have time for one more question.
Yeah, I would like to give the word to Corinne. I see now she cannot unmute, so I will look up her name and that I can unmute her, but I have to scroll to the list. Yeah, so Corinne, we can hear you now.
Okay, do you hear me now?
Yes.
But that was, I mean, in fact, that was on the previous protocol, Miriam. The question was about the follow-up and what if you collect cases of possibly COVID disease? So I understand that there is no, I would say, active follow-up. So the follow-up is at the discretion of the person who reports. Is that correct?
I think this was a question for Lynn. Do you want to address that, Lynn?
Yes, I'm not sure if I got your question right. So can you please repeat the question? It was about the follow-up.
With a follow-up, and especially if you have COVID disease reported.
Yes.
So there won't be any active follow-up. So you won't be able to do any estimation of the duration and the severity of the disease.
No, yeah, this is not really the aim of the project to look at the vaccine effectiveness. Huh? So yeah, I think this should be, it's important to discuss with the public health authorities in the different countries to see how you can monitor this. Yeah, of course, the vaccine effectiveness and vaccine failure is a bit on the interface of
I was more
monitoring
thinking
of the
about the vaccine-enhanced disease, Lynn, that's why.
Yes, yes. Yes, so this is something that is in between, so this makes it difficult. So our approach is not ideal to really study vaccine-enhanced disease. So yeah, so we will not actively follow this up, no.
Maybe just to comment on that further, Corinne, so I understand where you're coming from. So in the last webinar from VAC4U, we have discussed vaccine-enhanced disease and how you could monitor it, and we didn't come to a conclusion. We did come to a conclusion that needs further work, and so there is a working group established that actually will meet this afternoon to start thinking about it. So that can be the topic of a future consensus document or meeting. So for vaccine enhanced disease, we really need to see what can be done in an observational setting.
Thank you, Miriam.
Thank you. Okay, so then I would like to move on with the next presentation. So this presentation will be dealing with safety studies using electronic healthcare databases, and will be presented by my colleagues Caitlin Dodd and Corinne Milana.
Thank you, Miriam. Okay, next slide, please. Okay, as in the previous template protocol that was presented, here we intend to present a template for use by investigators for studying safety in electronic health records, and we provide a number of phases and designs for doing so. So here you see the schematic across the top, and moving from signal detection through rapid assessment of signals and through formal evaluation studies or formal association studies. So first, conducting signal detection in electronic health record data sources, we've outlined methods for identification of unknown adverse events following immunization using tree scan methodology, as well as rapid cycle analysis of pre-specified adverse events of special interest. And I'll go into some more detail on those in the following slides. For rapid assessment, so basically quickly evaluating detected signals using EHR, we describe ecological designs as well as unadjusted self-controlled designs. And finally, for full evaluation studies, we describe cohort, case control, and self-controlled designs that can be applied. Next slide, please. For signal detection, we provide a framework by which investigators can choose methods based upon the events they plan to study and the data available. So we describe tree scan methodology and rapid cycle analysis using maximized sequential probability ratio testing in electronic health record data sources. So tree scan methodology, and I will just describe these very briefly. Tree scan methodology basically it exploits the hierarchical structure of ontologies of coding systems that are used in electronic health records in order to identify clusters of events or groups of events that have a log likelihood that is greater than would be expected. So it's an observed to expected analysis, and it can be done using a self-controlled framework. Risk intervals can be varied. Basically, it identifies unforeseen adverse events that may need to be further studied. Now, rapid cycle analysis using maximized sequential probability ratio testing. Now, it's sequential probability ratio tests have been around for a long time. The maximized version of this, which is developed by Martin Kulldorff and others, uses a composite outcome, basically, in which a signal is flagged when the relative risk is greater than 1. And this can be done in many design frameworks, a self-controlled framework, also using historical or contemporaneous controls, which are depicted here. And the methods adjust for the fact that there are repeated looks at the data on a weekly or bi-weekly basis. Next slide, please. So the objectives for tree scan analysis are to assess whether there are sufficient observed unspecified events or groups of events, and this again is used using the ontology of the coding system to identify a potential safety signal, and that potential safety signal will then need to be assessed. Rapid cycle analysis using max SPRT uses rapid analysis to identify if there is a signal for a pre-specified adverse event of special interest following a specific vaccine product. Next slide, please. Now, moving on from signal detection to rapid assessment, we've outlined a couple of methods that can be used for rapidly assessing signals. One is using ecological designs, such as the interrupted time series design. And this can be applied when there is an intervention, such as a new vaccine, which will be the case in our situation, and comparing incidence of events before and after that intervention. This works best when coverage is high and the intervention can be very accurately identified at the start of the intervention. And this requires only data on cases and population denominators. Additionally, for rapid assessment, one could consider using an unadjusted self-controlled risk interval. This could be applicable if an investigator doesn't want to wait for post-risk window time to accrue, and they can therefore use a control window that is before the vaccine. Care must be taken if the event of interest is a contraindication to vaccine, or if it delays vaccination, and the investigator can take this into account by identifying a healthy vaccinee period to remove from the control period. But as is indicated in the name, this is unadjusted for time varying covariates, but can provide results rapidly. Next slide, please. So the objectives of ecological analysis, including interrupted time series, is to assess whether there's increased incidence or a change in incidence of a pre-specified severe adverse event following introduction of a COVID-19 vaccine product in an identifiable population. And the objective of an unadjusted self-control risk interval is to quickly determine whether there's increased incidence of a pre-specified adverse event following exposure to a COVID-19 vaccine product, as compared specifically to a pre-vaccination control period. Next slide, please. At this point, I would like to hand over to my co-author, Corinne, to discuss signal evaluation. Thank you.
Thank you, Kathleen. So I will briefly present the study designs. Yes, thank you. I will briefly present the study designs that could be implemented for signal evaluation once a signal is detected or when there is an identified or a potential risk observed during the development of the vaccines product. We classified those designs as first the traditional ones, which include cohort and case control design. In the cohort design, what you will do is to compare the incidence rate of the event under assessment between an unexposed cohort, then exposed cohort, which is exposed, subject exposed to the vaccines under assessment, and you will compare to an unexposed cohort, which can be a concurrent unexposed cohort or an historical unexposed cohort. You can also consider to have those two types of unexposed cohort in your study design. There are here several points that are important to consider for this cohort design. The first one that I would like to highlight here is the confounding factor. And the use of appropriate methods for the adjustment for those confounders. Adjustment can be performed at the time of study entry at time 0. For example, by using frequency matching or propensity score matching, or adjustment can also be performed at the time of the analysis. And a second important point to consider is the follow-up time and the observation period, which will depend first on the outcome that is under assessment, but also that will depend on the recommended vaccination schedule. Second type of design, traditional one, it's the case control. So here you will compare the cases of case of a specific event versus controls that are matched for at least the main confounding factor, and you will look back in time if those cases and control were exposed or not to the vaccine under assessment. So next slide, please. So here it's a brief overview of the self-control design. I will not go into detail because it has already been explained before. So there is the most famous one, which is the self-control case series. where you have a pre-specified risk period that occurs just after your exposure, and then you have your control period, which usually correspond to the remaining observation time in your study. And then there are two variants of this method. There is the self-control risk interval and the case crossover. For the self-control risk interval, you have a pre-specified risk period and a pre-specified control period, and you compare the incidence rate between both periods. For the case crossover, this design considers the occurrence of the event as fixed and the exposure to vaccine random. Those designs are usually more suitable for adverse events with an acute onset and a short latency. So next slide, please. So the objective for this type of design for signal evaluation would be to address whether there is an increased risk of pre-specified severe adverse events following vaccination with a specific COVID-19 vaccine product. And another objective could be to assess whether there is an increased risk of pre-specified severe adverse events following vaccination with specific vaccine groups defined by platform and or component. So next slide. So in our template protocol, we also provide some guidance about the data requirement for using electronic health records. We classify those requirements in two types. So first is the data element, which includes population denominators, the need for information on events, on covariates, and on exposures. And the second type is the characteristic of the data source, for which we consider that weekly updates would be more appropriate, especially to conduct a signal detection assessment using the MaxSPRT method. And also a second characteristic would be the availability of data for controls, which obviously is not applicable for self-control designed. So next slide, please. So I will finish with the limitation for the different type of study design that we can apply in using EHR. So there are limitations related to the use of secondary data. There are always questions about accuracy of medical codes and algorithms, and there is the risk of misclassification of the outcome. Also, questions about validity of the exposure and availability of brand, vaccine brands, which it's unknown today. Also, more general, there is limitation related to the fact that absence of records does not imply the absence of an event. Another limitation is the use of the same data source for signal detection and signal evaluation that we can further discuss. And then related to specifically to the study design for signal detection for the TRISCAN methods, the analysis depends really on the hierarchical structure of the ontology of the coding system that is used in the data source, which may not always be favorable to identify clinically relevant events. For ecological designs, they may have the presence of concurrently time-varying changes in the post-vaccination period, and those factors may impact the incidence rate that you will observe in the post-period versus the pre-vaccination period. And for limitation for signal evaluation and the study design for signal evaluation, we wanted here to highlight the bias related to immortal time, especially when we want to assess the pregnancy outcomes. So there are methods to help with that, so like time-dependent covariates and or time-dependent exposure. Also adjustment for confounding factor, so or at time of study entry or during the analysis. And also for the self-control design, it's really the duration of the washout period that could be sometimes longer, and they may have a risk of changes in behaviors over time, especially for subject with comorbidities and maybe change of medication, use of medication over time. And that's the end for me. Thank you. If
Thank
you
you
have
very
any
much, Colin and Caitlin. Yeah. So just because we have 3 minutes left and we have a lot of questions. So before we allow maybe 1 or 2 questions, I would just like to wrap this up by this is like how this goes forward. So what we have presented today will be part of 3 different template protocols that will be submitted to EMA and then will be reviewed. Once they have been agreed by EMA, they will be made publicly available. But currently they are not yet. But these are the proposed methods. So any questions you have, please reach out to the secretariat at vac4u.org. We know, Kat, you had your hand up, so for questions. So I would say Kat Bolat.
Oh, yeah. Well, it was actually at the very beginning for the intensive monitoring. I'm just wondering, do you think that the added value of such a system would be on top of the data that's being collected in the phase 3 studies? Because these will also be large studies, carefully monitoring safety.
Yes, that's a good question, and I have that question myself, especially considering that it's not advisable. Sorry,
this was for the intensive monitoring.
Oh, my apologies. I thought you meant the beginning of our presentation. Sorry.
No worries, Caitlin. So for Lynn, I guess.
Yes, I understand the question. So yeah, one difference could be, for example, the target group for vaccination might be different once the vaccine are on the market, for example. This would be one thing to keep an eye on. Right? Yeah. Also, I'm not sure for each vaccine how long the follow-up periods are now in the clinical trials.
Yeah, that was my second question. Like, what's the duration of follow-up, or you aim for?
Yes. So now we define the minimum set of follow-up periods, so that every country will have at least those follow-up periods. But I know, for example, there are countries that now have the latest follow-up at 1 year after vaccination.
And then I have a question. I think it's for Corinne, I think. I mean, why would you use an historical cohort for signal evaluation?
So yes, that's an approach that could be used maybe if you want to have a population which is totally naive with regard to the SARS-CoV-2 virus. But yeah, we need to think about the usefulness of this. Yes.
Yeah, and then I agree. And then my other question was like, when do you have different designs listed, but given our knowledge of the epidemic, I mean, is there some designs that you think should be preferentially used? Like, one of my main concerns, for instance, is that vaccinated and the unvaccinated populations will be very different, as we are likely to vaccinate the risk groups first.
Yeah, but yeah, I think it's really the fact that you need to have a good adjustment, and I was mentioning the propensity score matching at the time of study entry, that it can help you to control for any major confounders. Yeah. And then it depends on the objective. Yes, for the follow-up time, it depends on the event that is under assessment. Yeah.
And would you be able to formulate any guidance on this? Like, for this type of event with this type of characteristics, we would recommend to use this design, and otherwise better go for another design?
I think we
now you list possible designs without recommendations when to use what.
Yes, that's true. We could, but the list of ASI is very, very long, and yeah.
But I was thinking of more like at a higher level, characteristics of the adverse events, like.
Yes, but we have in the protocol template, we have included some criteria to help to decide which type of design is more suitable. Yes.
Okay, thanks.
Yes.
Okay, I think The time for the webinar is over. There are a lot of remaining questions for sure, and there are questions in the chat or in the questions. We will get back to you. Is there, Vincent, you had also for a long time a question, so please, we give you the last chance as the founder of Vak4U, co-founder.
Can you hear me now?
Yes.
No, I have a few questions, and I've tried to put some in the chat, or I was not sure I was in the right chat, but anyway, maybe you will see my questions. But maybe one question now for the LAREP again, the intense monitoring. So a very good point about this is that it will be real time, as I understand, using apps. But it's a signal detection study in my view. I think that's also what you said, Miriam. But what is not clear to me is how the signals will be identified, because basically it's not by measuring incidence rate that you can say that something is a signal or not alone.
Yes, so that's correct. We'll be looking at the patterns of AFIs and course of disease. This is more the aim of the intensive monitoring. Yes, so yeah, the signals will be more like this. It will not be like there'll be a limit of detection for a signal, but we'll try to describe the patterns of AFIs and the course of disease, the impact.
So you mean it will be more like an expert medical assessment of what the data will show?
I'm not sure if I understood that correctly. Can you explain a bit more, please?
Yeah, but if I understand what you've said is that you will look at the patterns, not in a statistical way. You will look at that with relevant medical expertise. Is that what you mean?
No, we can look at it in a statistical way. For example, we can still compare frequencies between different vaccines, for example, let's say, or between yeah, people with different
vaccines. Comparing different vaccines is a bit risky because first, the vaccines are very likely to be used in different countries and also at different time and different risk groups. So you will have to adjust for a number of factors or restrict to vaccines used in the same country or in the same time or the same risk group, which will, I think, limit your power. So comparing vaccine, I would think twice about what you can actually obtain as robust comparison from this.
Yes, yeah, for sure we have to take confounding factors into account. Yes, I agree. Yeah, but yeah, okay. But still we can do a statistical analysis. Yeah, I would say. Yeah.
Okay, thank you. I think when we have the protocol, this will be probably clearer to us.
Yeah.
Thank you.
Well, thank you, everybody. So I think it's time to finish or to close this session. I would like to thank everybody who has been joining on behalf of Vak4U, and thank you for the great questions and presentations. And again, those of you who have questions in the questions, you will be emailed. We will get back to you. Thank you very much.